Hello everyone, welcome to PW English YouTube channel. I'm Shalini Somashekhar, your botany teacher, and in today's lecture in the UMEED NEET English batch, I'm going to be discussing the chapter Biological Classification. You might already know that in this batch, we're going to be discussing every chapter in absolute detail in one shot. Before we start the discussion of the chapter Biological Classification, let's quickly go through the number of questions that have been asked from this chapter over the past few years. If you take a look at this table or the analysis that we have made, you can see how consistently from the year 2017 to the year 2024, one question has been asked from this chapter, and in majority of the cases, these questions are example based. You will be learning a lot of examples for a lot of different groups in this chapter, so you can expect a question from those examples, all right, except for the year 2023 where no questions were asked from this chapter. Majority of the cases from 2017 you have one question, and in 2016 2 to 3 questions also have been asked. This does not necessarily mean that in the upcoming NEET 2025 exam there will be just one question. It could change, it could be one, it could be two, it could be anything. We should be the best prepared, so we're trying to prepare you for the best. We will learn all of the concepts perfectly so that no matter how many questions from whichever corner of NCERT is asked, you should be well prepared to answer it effortlessly, right? That's the whole goal of this batch. So let's get started. Talking about the syllabus, in this chapter, all of the concepts are included in the NEET syllabus. You can either refer your old NCERT or new NCERT, nothing is deleted. So this is a snip of the NEET 2025 syllabus that is posted on both the NTA and the NMC websites. You can take a look at it. So we are going to be learning about the 5 kingdom classification, salient features and classification of the 5 kingdoms, that is Monera, Protista, fungi and lichens, viruses, viroids, prions and all of that, okay? So we learn all about it in a lot of detail. So let's get started. If you remember, in the previous chapter, we spoke about the need for classification and we spoke about how classification happens, what are the different taxonomic categories. I gave you the example of how there will be many levels in classification by using library for analogy, right? And how the books are arranged in a library. If you've gone through the lecture for the previous chapter, you would definitely remember. So in this chapter, as the name itself suggests, we are going to learn about how classification of living organisms have happened. What are the different systems of classification? How have the criteria for classification changed and evolved with time? We're going to look into all of that. Okay, so let's start and understand how classification was done very early in history. Okay, so some of the earlier systems of classification did not have a scientific basis. For example, very early on, you know, humans and primates, we're very inquisitive beings, we're very curious beings. We want to understand what is happening around us and we want to try and make use of whatever we can from nature, right? So even our ancestors very early on, they were curious about nature and they wanted to know how they can make use of different living organisms for their benefit. So back in the day, classification used to happen, but it was not a very scientific way of classifying living organisms. It was done mostly instinctively. Instinctively is the word that is used in NCERT. Now, what does instinctively mean? Instinctively means, you know, they say gut feeling, right? You don't put much conscious thought into it, you just do it, okay? So, very similarly, even living organisms, when they were being classified, there was no scientific basis or criterion behind it. It was just classified instinctively, mostly based on whether that organism could be used by humans or not, whether it was useful or not. Useful in what sense? Whether it could be used as food. If some mushroom that you're looking at, is it poisonous that could kill them, or it is something nutritious and delicious as well. So that is one way of classifying things, based on whether it can be used as food or not, based on whether something can be used as a material for clothing or not, based on whether something can be used for shelter or not. So based on how living organisms that were found around human beings, based on its use for us, our early ancestors classified them into different categories. But one of the earliest systems of classification which was kind of scientific was Aristotle's classification. Everybody knows about Aristotle, I guess. Aristotle attempted to classify both plants and animals in various ways. Two examples are given to you in your NCERT textbooks. First, let's talk about how Aristotle classified plants. It was very simple. The criterion for classification was very simple, but it was scientific. How it is different from the early classification, which was not scientific at all, which was done instinctively, is that Aristotle's classification, though it is simple, it was scientific. How did he decide to classify plants? He used just one basic criterion, which is morphology of the plants. He just looked at the plants. What is morphology? As biology students, I'm sure you know that morphological characters or morphology means basically we are talking about external features. Okay, so you don't need any fancy microscopes to, study the morphology. With your naked eye, whatever external features you can study, that is morphological studies, right? So, how he classified plants was based on morphological features. So, he classified plants into 3 different groups. It might come across as a very silly thing, but back in the day, people did not have much scientific temperament. They did not think about it in terms of science. So at that time, this was very significant because earlier than this, people did not use science at all for classification. Today, a second grade child, a third grade child would be very easily able to tell the difference between a herb, a shrub and a tree, but back then it was significant, right? So how did he classify living plants? He classified plants into 3 different categories, that is herbs, shrubs and trees. This is very simple, right? So something like a short plant which has kind of a green flexible stem, you can call it a herb. It doesn't grow very tall, it doesn't become too bushy, that is called a herb. Now what is a shrub? Shrub is slightly taller than a herb. It could have woody stems, it could have a lot of branching, but it will not have like a proper trunk similar to a tree. So that is your shrub. Trees are the biggest type of plants. So trees have a solid thick trunk, it has branching and all of that. So morphologically, you just look at it with your naked eye, you will be able to differentiate between these 3 types of plants, right? Similarly, he attempted to classify animals as well. When he classified animals, he used just one criterion. That is whether that animal's body has red colored blood or red color blood was not there. Whether the blood in that organism was red in color or not. That is the only criterion that he used. Okay, so he classified animals into 2 groups. One group that did not have red blood, he calls it anema. And the other group, like us, vertebrates, which had red colored blood with hemoglobin. That is anema. Which are some organisms that do not have red colored blood? Like insects. If you've ever killed a cockroach, you would know that the fluid, the blood inside cockroach is not red like our blood. It's colorless, right? It's like somewhat whitish, colorless kind of blood. If you've ever squished a mosquito between your hands, between your palms, you might have seen some red color blood. But mosquito is also an insect. The red color blood that you see while you kill a mosquito is not really the mosquito's blood, it's the blood of us that the mosquito would have sucked. So there are organisms like insects which lack a red colored blood, and there are organisms like us which do have red colored blood. Based on this simple criterion, he tried to group animals into enema and anema. Okay, now let's move on and learn about the 2 kingdom classification of Linnaeus. Who is Linnaeus? Have we heard about him before? Yes, we have. In the previous chapter when we spoke about binomial nomenclature, I have told you that Linnaeus was called the father of taxonomy. He's the one who, published Systema Naturae. He's the one to have proposed the binomial nomenclature and all of that, right? It's the same guy. His name is Carolus Linnaeus. Some people call him Carl Linnaeus or simply Linnaeus. So he is the first person to have grouped living organisms into 2 distinct groups, 2 distinct categories or 2 distinct kingdoms. Now, what is a kingdom here? Kingdom is one of the taxonomic categories we've learnt about in the previous chapter. Remember, we learnt about kingdom, phylum, class, order, family, genus, species, all of that. So it's the same kingdom that we're talking about here. It's a group of organisms that have some similarities, okay? So, in fact, it is the highest category in all of your taxonomic categories, it includes a lot of organisms. So Linnaeus used just one simple criterion to classify all living organisms. The one simple criterion that he used was whether cell wall was present or absent. Presence or absence of cell wall. Regardless of what the cell wall was composed of, all he thought about was one criterion, and that one criterion is whether that organism has a cell wall or it doesn't. If the organism had a cell wall, he would group it into kingdom Plantae. If that organism lacked a cell wall, he would group that into kingdom Animalia. Alright? So these two kingdoms, kingdom Plantae and kingdom Animalia, the primary difference according to Linnaeus for classification is whichever organisms possess a cell wall, they would be grouped under kingdom Plantae, and those organisms that lacked a cell wall, they would be grouped under kingdom Animalia. Alright? Now, this two kingdom classification was brilliant back then. But as and when more and more organisms started to get discovered, as and when microscopy started to get advanced and we got to know about a lot more species of microbes, we started to see some disadvantages or drawbacks of the system also. One thing is, it's very easy to understand. Anybody can understand this classification. That is one advantage of this classification. It's not very scientific, so you don't need to know a lot of anatomy, morphology to understand this, because we're looking at just one simple criterion. Does it have a cell wall or it doesn't? If it has, it's a plant. If it doesn't, it's an animal. But that was an advantage, that it was easy. It was very easy to do, and it was very easy for anybody to understand. But it had certain disadvantages. In fact, there are three very important disadvantages to the two kingdom classification. As and when newer and newer organisms started to get discovered, they got to know that some of these organisms did not fit into either category. So, yeah, we learn more about the disadvantages. Firstly, in this two kingdom classification, there was no clear distinction between eukaryotic and prokaryotic organisms. You know structurally how prokaryotes and eukaryotes are very different from each other, right? Prokaryotes are very primitive as compared to eukaryotes. Despite being so different from each other, there was no attempt made to distinguish these two kinds of organisms. The bacteria that were prokaryotic were grouped with algae that are eukaryotic just because they both had a cell wall, right? So that is the first disadvantage of the two kingdom classification proposed by Linnaeus. Bacteria and cyanobacteria, both of them are prokaryotic. Bacteria and cyanobacteria, both of these are prokaryotic. They were grouped with higher plants such as algae, bryophytes, pteridophytes, all of those, okay? Just because they have a common cell wall. But if you think about it, even the cell wall material is not the same. In bacteria, the cell wall is made up of peptidoglycan, while in plants it is made up of primarily cellulose. There also there is a difference, but it was not taken into consideration. Now the second thing is, unicellular organisms were grouped along with multicellular organisms. There was no distinction between that. Chlamydomonas and Spirogyra, these are unicellular. Algae, many of them are multicellular. So there was no distinction between unicellular and multicellular organisms. That is the second disadvantage. The third disadvantage is it did not take into consideration the mode of nutrition. Now, if you look at the plant kingdom, according to the two kingdom classification, everything that has a cell wall has to be grouped in that. If you look at bacteria, they are prokaryotic but have a peptidoglycan cell wall. Next, if you look at algae, they are eukaryotic but they have a cellulosic cell wall. Finally, look at fungi. Fungi have a cell wall that is made up of chitin and they are not autotrophic, they are heterotrophic, right? So, very diverse group of organisms were put in the same kingdom just because they all have cell walls, right? So, the other thing is autotrophic organisms as well as heterotrophic organisms or saprotrophic organisms like fungi were put under the same group just because they had a cell wall. Okay, so over here, photosynthetic organisms are the plants, like you already said. Non-photosynthetic organism is the fungi. Fungi that had chitin in their cell walls, and autotrophic algae and plants that had cellulose in their cell walls, they were grouped together. So these are some disadvantages. Another thing is, some organisms, like I already mentioned, they did not fit in either category. Let's take the example of euglena and slime molds. Everybody knows euglena, right? You've learnt about it in your lower grades as well. It kind of looks like this, right? So it will have a nucleus, and there's something that it has called an eye spot. This is euglena. Okay, so this euglena is unicellular. It is eukaryotic. It can be photosynthetic like plants, or it can be holozoic like animals, and that depends on the availability of light. Okay, like plants, this euglena will have chloroplast. That means it has the ability to perform photosynthesis. Now for photosynthesis, chloroplast is essential along with chloroplast and pigments to trap the energy present in light. Sunlight is required, right? It's an absolute requirement for photosynthesis. Whenever light is available, this organism, euglena, will make use of the sunlight, trap the energy by with the help of the pigments present in chloroplast, and cook food, make glucose by photosynthesis, and obtain this nourishment from in that way. But sometimes when there is not sufficient light available, then it will turn like an animal. It will start engulfing smaller organisms and ingesting those organisms like how we animals do. In fact, this has this eye spot which can kind of sense where light is available, and with the help of the flagella that are present very close to it, it will draw the organism or pull the organism towards the source of sunlight so it can photosynthesize. But let's say there is no light available at all, then it will it will engulf smaller organisms and obtain its nourishment like that. Now, depending upon the presence of sunlight, this can either be autotrophic like plants or it can be heterotrophic like animals. Now, in linear system of classification, in the two kingdom classification, you know that in order to call an organism, include an organism under plant kingdom, it should possess a cell wall. But this euglena does not have a cell wall like plants. It has a proteinaceous covering outside which is called the pellicle. It's kind of flexible. Okay, so this is photosynthesizing like plants when light is available. It does not have cell wall like plants. It has a covering that is made up of protein, which you don't find in plants or animals. And sometimes when light is not available, it's even eating food like animals, right? So where do you put this in? Do you put this under plant kingdom or animal kingdom? That is a confusion. And then we have another group of organisms called slime molds. These are super intelligent, very tiny organisms. Like, though they are primitive, they are very smart. So these slime molds, in different phases of its life, how the cells are will change. Okay, so there is one phase in its life called the amoeboid phase, where the cells don't have a cell wall. But whenever unfavorable conditions are there, it will form a fruiting body. Within the fruiting body, spores will form, and those spores will have a cell wall. If you look at the mode of nutrition of slime molds, they are like fungi. They are saprotrophic or saprophytic, you can say. They obtain their nourishment from dead and decaying organisms, which is very much like fungi. But whenever they develop cell walls in the spores, the cell wall is composed of cellulose, which is like plant cell wall. So where do you put this in? Where do you group this organism? In one phase of its life cycle, it doesn't have a cell wall. In another phase, it has a cell wall. When it doesn't have cell wall, you put it in Animalia, and when it has a cell wall, you can't put the same organism in Plantae, right? Because it's one phase where there is no cell wall and one phase where there is cell wall. Both of them are part of the same organism's life cycle. You can't put the same organism in two different groups. If this has shared characters of plants and animals in terms of possessing cell walls, where do you put such an organism? That is also confusing. So this system though it was scientific, though it was helpful, it was found to be inadequate. Inadequate means not sufficient. In general English, it was not sufficient because over here we used morphological features and just one morphological feature primarily, which is the presence or absence of cell wall. That was not sufficient because you had a lot of confusions and disadvantages to this kingdom. So later they realized that in order to classify organisms better, you need to study the organisms more. You have to look at its cell structure, whether it's prokaryotic, eukaryotic, whether it is unicellular, multicellular, what is its mode of nutrition, what are the chemicals that are present inside of it, what are the evolutionary relationships it shares with other groups or other organisms. So all of this was taken into consideration and the classification systems evolved with time. Okay, so besides gross morphology, scientists and taxonomists felt that you should use cell structure, prokaryotic or eukaryotic, nature of wall, whether it was made up of peptidoglycan or cellulose or chitin, what was it composed of, mode of nutrition, whether it was autotrophic, heterotrophic, if it was heterotrophic, how would it obtain nutrition, habitat, where does it live, how does it reproduce, asexually, sexually, what kind of spores are produced, evolutionary relationships. They started using information from all of these branches of biology also while classifying living organisms. Similar to how you have updates on your mobile phones or electronic gadgets, well, when they know that something isn't working, they will upgrade that, they will make certain fixes and upgrade that whenever there are bugs and stuff, right? So very similarly, even in science, even in classification, taxonomy, at that time, whatever knowledge we have, using that knowledge, we do classification. As our knowledge gets updated, as we will learn more and more things about living organisms, we use that knowledge to better the classification system so that there is as little confusion as possible in grouping organisms. All right. So after the two kingdom classification that was proposed by Linnaeus, there was a three kingdom classification that was proposed by Ernst Haeckel. In all the systems of classification, remember, we are going to study about, we already studied about the two kingdom classification. Going forward, we're learning about the three kingdom, four kingdom, five kingdom, six kingdom classification as well. In all of these different kingdoms, different systems of classification, what remains constant is Plantae and Animalia. Plantae and Animalia are going to stay throughout all the systems of classification. Along with that, newer systems will get added. So in the three kingdom classification proposed by Haeckel, the other extra kingdom that got added was kingdom Protista. Okay, just remember this. Ernst Haeckel, three kingdom classification, Plantae Animalia is always going to be there. Along with that, there is Protista. More about Protista I'll tell you going forward. Okay, and then after this, there was a four kingdom classification proposed, which was done by Copeland. And along with Plantae, Animalia, Protista, we now have Monera. Okay, so this is the four kingdom classification. And after this came R.H. Whittaker's five kingdom classification, which is the most widely accepted and currently used system for classification. So in R.H. Whittaker's five kingdom classification system, they've used criteria from diverse fields of biology. We will look at its cellular structure, whether it is prokaryotic or eukaryotic. Body organization, whether it is unicellular or multicellular. Mode of nutrition, whether it is autotrophic or heterotrophic. Reproduction, how does it reproduce asexually, how does it reproduce sexually, what kind of spores are formed, based on all of that. Phylogenetic relationships is basically the evolutionary history of that particular organism. How closely they are connected with other species, things like that. If you consider us and a primate, another primate like a gorilla and a dog, you know that we're more closely related to dogs, more closely related to gorilla than we are to dogs. So that makes it easier for grouping us into the same group as, primates as opposed to dogs. So basically, because we evolved from apes, we know that we are closely related to them, we share more similarities, and it's easier to group us into a common bag, right? So evolutionary relationships plays an important role in classification. More about it as we move on, you will understand. Okay, so in R.H. Whitaker's 5 kingdom classification, you have 5 kingdoms: Monera, Protista, fungi, plantae, and animalia. Okay, so most people know the order as Monera, Protista, fungi, plantae, and animalia. But when they ask you in the 3 kingdom classification, which was the additional kingdom that was added, people will think it's Monera because Monera comes first, but that's not the case. It was Protista. And then in the 4 kingdom classification, which additional kingdom was added as opposed to 3 kingdom? It was Monera. And fungi is the kingdom that got added to the 4 kingdom classification in the 5 kingdom classification. Okay, I hope it is clear up until this point. Now let's understand how R.H. Whitaker classified living organisms into 5 different kingdoms. All right. Now, let's start with all living organisms. Okay, every organism that has been discovered, described so far, we'll include them all. And let me write here living organisms. We are going to use all of these criteria that I had mentioned one by one. The first criterion that we are going to use is whether the organism is prokaryotic or eukaryotic. I guess you already know what prokaryotic and eukaryotic means. They are 2 different types of cells. Prokaryotic cells are those cells in which the genetic material is not covered by a nuclear membrane, and eukaryotes are those cells, or eukaryotic organisms are those organisms in which the cells have DNA or a nucleus that is covered by a nuclear membrane. Whether the nuclear membrane is present or absent is what differentiates prokaryotes from eukaryotes. Now, what are we looking at? The first thing, you're looking at the organism, you're looking at its cell under the microscope, and you're checking for whether the nucleus within the cell is surrounded by a nuclear membrane, whether the genetic material is surrounded by a nuclear membrane or not. If it is surrounded by a nuclear membrane, it is prokaryotic. If it isn't, then it is eukaryotic. Okay. So the first question that we're going to look at here, or the first criterion you can say, is whether it is prokaryotic or eukaryotic. Right? So it could be prokaryotic or eukaryotic. If it is prokaryotic, then we are going to include that under kingdom Monera. We will include that under kingdom Monera. Now what are we left with? We are left with eukaryotic organisms. Right now, we will look at the second question here, whether it is unicellular or multicellular. If it is eukaryotic, it could be unicellular or it could be multicellular, right? So, we will check that. This can be unicellular or it can be multicellular. If it is eukaryotic and it is made up of just one single cell, if it is unicellular, we will group that in kingdom Protista. Two kingdoms done. What are we left with? We are left with eukaryotes that are multicellular. What is the next question that we will ask? What is its mode of nutrition? Whether it is autotrophic or heterotrophic. Okay. These multicellular eukaryotes can either be autotrophic or heterotrophic. Some of them are photoautotrophic, some of them show absorptive heterotrophy, some of them show ingestive heterotrophy. Okay. Now, whichever one shows photoautotrophy, those organisms are grouped under kingdom Plantae. Those organisms that show absorptive heterotrophy are grouped under kingdom fungi. The organisms that are grouped, the organisms that show ingestive heterotrophy is grouped under kingdom Animalia. So right here we have 5 different kingdoms, right? Here we have the first kingdom, here you have the second kingdom, then you have the third, the fourth and the fifth. Let's now understand the various modes of nutrition that we see in these 3 groups, okay? You know what is auto? Auto means self. Autotrophic means capable of synthesizing food by themselves. They don't depend on other organisms to synthesize food. However, they depend on light for synthesizing food. So we call it photoautotrophic. Photo is light, autotrophic is capable of making food by themselves. We know all plants will have chloroplast within which pigments like chlorophyll will be present. Those pigments trap the energy present in sunlight and convert that into use that to synthesize organic matter like glucose, right? So photosynthesis is performed in these organisms. That's why they're capable of synthesizing food by themselves. So all of the plants would be included here. Now what does absorptive heterotrophy means, or what is heterotrophy? Heterotrophy is organisms not capable of synthesizing their own food. They depend on other organisms for synthesizing, for their nourishment. They do not have the ability to make food by themselves, so they depend on other organisms for nourishment. That is heterotrophy. Under heterotrophy, you have many different kinds. Okay, for now, just understand the difference between absorptive heterotrophy that we see in fungi and ingestive heterotrophy that we see in animals like us. What is absorptive heterotrophy? Heterotrophy is it cannot synthesize its own food. It obtains its food by absorbing nutrients from its surroundings. See how fungi grow. Where do fungi grow? Fungi grow on dead and decaying matter mostly, right? So how does it obtain its nourishment? Fungi, the body of the fungus is very, very simple. You do not have different types of tissues found there. It's just like a filamentous structure that is aggregated and formed like a network, right? So you see hyphae and mycelium. More about it we'll learn moving forward. For now, just understand. Fungi cells are the structure is extremely simple. You have many cells lining, I mean, lying one on top of another, it forms a filament like that. It will show some branching, and it will form a network. That is basically the structure of fungi. This does not have an elaborate digestive system, a mouth or anything. So how does it obtain nourishment? It cannot synthesize anything by itself. How does it obtain nourishment from other organisms? By absorbing nutrients from the outside, right? So if, let's say, there is some dead leaf or some dead organic matter present in an area where this fungus is growing. Now this fungus will produce enzymes, hydrolytic enzymes inside its body, release it to the outside. Now these enzymes will act upon this dead and decaying matter and convert that into inorganic substances. The fungus cannot absorb the leaf as a whole. It will secrete some enzymes outside. Those enzymes will break down the organic matter present around it. As a product of that breakdown, certain very simple organic molecules will form. Those simple inorganic molecules, not organic, those simple inorganic molecules are absorbed by the fungus. Okay, so that is why we call it absorptive heterotrophy. Simple molecules are formed, they could be organic, inorganic. Okay, so more about it as we move on, you'll understand. For now, just remember why we are calling this absorptive heterotrophy is because they themselves are not capable of synthesizing food. They absorb nutrients from its surroundings. How can they absorb it? If a leaf is fallen down, if a dead animal body is there, it cannot absorb it as a whole, right? Also, if there are big molecules, it cannot be absorbed through the cell membrane. So therefore, they will produce certain enzymes that can break those big molecules down. The breakdown products will be smaller molecules that are capable of being absorbed. It will absorb that and use those molecules as a source of nourishment. Now, why do we call this as ingestive heterotrophy? What does that mean? Ingestion is taking in food into the body. If you think of us, we also have enzymes that are capable of, capable of breaking down food, right? That is what our digestive system, different parts secrete. In our mouth, there is an enzyme that is secreted that can break down starch considerably. Our stomach releases certain enzymes, our intestines, pancreas, all of these produce enzymes, right? So what is the function of these enzymes? Whatever fungus is doing outside, we are doing within the body. Fungus will not take its food inside its body. It will secrete the enzymes outside. What do? We take in food inside the body. The same kind of enzymes are secreted in different parts of our digestive tract. It does the same job of breaking down big molecules and converting them into smaller molecules. And these smaller molecules will eventually get absorbed into the blood and get transported throughout the body, right? In animals, that is what happens. So over here, we ingest the food. Enzymes are secreted, it gets broken down, and that is how we obtain nourishment. So the mode of nutrition here is ingestive heterotrophy. We cannot synthesize food by ourselves. We will ingest it, break it down, and then utilize it, right? So that is ingestive heterotrophy. Let's go through this one more time. All living organisms are considered. The first question that we are asking is whether it has a membrane around the nucleus or not. If it doesn't have, we call it prokaryotic. All organisms that are prokaryotic are grouped under the kingdom Monera. Next question. What are we left with first? We are left with all eukaryotes. What are we going to consider for classification of eukaryotes further? The number of cells. Does it have a single cell? It is made up of a single cell, or is it made up of multiple cells? If it is made up of a single cell, if it is unicellular, then you will include that under kingdom Protista. So in kingdom Protista, you have unicellular eukaryotic organisms. Now what are we left with? We are left with multicellular eukaryotes. What are these multicellular eukaryotes? How do we classify them further? By looking at their mode of nutrition. Is it autotrophic? Is it absorptive heterotrophic? Is it ingestive heterotrophic, right? So looking at these criterion, we can classify them into kingdom Plantae, kingdom Fungi, and kingdom Animalia respectively. So this is the 5 kingdom classification. If you noticed, we had included criterion like reproduction and phylogenetic relationships and all of that, right? But over here, for this kingdom level classification, we are not utilizing it. As we move on, we will learn about how fungi are further classified. Over there, we will use reproduction and further classification. Phylogeny is also considered. Okay, for now, we have asked only 3 questions for grouping all living organisms into 5 different kingdoms. If you've understood, let me know in the comments. I will be very happy. Okay, now let's move on. And we have to learn about all of these kingdoms, like mostly kingdom Monera, Protista and kingdom Fungi in detail. Kingdom Plantae and kingdom Animalia, there are exclusive chapters for that called Plant Kingdom and Animal Kingdom. More details about these two you will learn in separate chapters. In this chapter, you will learn about Monera, Protista and Fungi in absolute detail. Okay, now this is a table that is given to you in NCERT. Also remember, R.H. Whittaker was the person who proposed the 5 kingdom classification in 1969. Okay, remember the date as well, because it's mentioned in NCERT. Now, what are the characteristics? We are looking at the cell type, nuclear membrane is there or not, organization of the body, whether it's unicellular, multicellular, does it have a cell wall, what is its mode of nutrition, all of that. Okay. So, first thing, if nuclear membrane is absent, obviously it will be prokaryotic, right? So, if it is prokaryotic, it is grouped under kingdom Monera. If the nuclear membrane is present, it's eukaryotic, it will belong to the other groups. In Protista, Fungi, Plantae, Animalia, we have eukaryotic organisms. Is it unicellular or multicellular? All prokaryotic organisms are unicellular. In Protista, we exclusively have organisms that are unicellular and eukaryotic, right? And fungi, plants and animals, you see multicellular organization. Next, in Monera, whether cell wall is present or not, cell wall is there, but it is not made up of cellulose like plant cells, it's made up of peptidoglycan, which has polysaccharides and amino acid in it. In Protista, some organisms have cell walls, some don't have cell walls, so it's a mixed bag. In fungi, cell wall is present, it's made up of chitin. In plant, cell wall is present, it's primarily made up of cellulose. In animal, cell wall is entirely absent. Monera and Protista, you have both autotrophic and heterotrophic organisms. In kingdom Plantae, you have all autotrophic organisms. In Animalia and fungi, you have all heterotrophic organisms. Okay, so this is something easily you can understand and remember, right? Now, let's talk about the 6 kingdom classification or the 3-domain classification that was proposed by a person called Carl Woese. Okay, what is this 3-domain classification? So he classified all living organisms into 3 different domains. The 3 domains are domain Archaea, domain Bacteria and domain Eukarya. Remember, the 5 kingdoms that we know of are Monera, Protista, Fungi, Plantae and Animalia. What he did was, he included all the eukaryotic kingdoms into a domain that he calls Eukarya. So within domain Eukarya, you have 4 kingdoms present, which are the eukaryotic kingdoms, Protista, Fungi, Plantae and Animalia. And he splits Monera into 2 domains. One is domain Archaea, which includes Archaebacteria, and the other one is domain Bacteria, which includes Eubacteria. So basically, how did we get the 6 kingdoms? Monera was split into 2 kingdoms, all of the other kingdoms remain the same. Okay, so this is called the 3-domain classification. You don't have to know much about it, just know which are the 6 kingdoms, which are the 3 domains, that's sufficient. Okay, so what are we going to do next? We will start learning about each kingdom of the 5 kingdom classification, starting with Monera. Now, what do we already know about Monera? We know that Monera includes prokaryotic organisms, and prokaryotic organisms are organisms that do not have a well-defined nucleus, they lack a membrane-bound nucleus, and what we also know is that they are unicellular. So, in Monera, the sole members are our bacteria. You know, there are many different kinds of microorganisms. Microorganisms are those organisms which you cannot see with your naked eye. They are too tiny to be visible to our naked eye. If you have to look at it, you will have to look at it under the microscope, right? So there are many different types of microbes. You have bacteria, you have protozoans, you have viruses, you have many different kinds of, microorganisms. Of all the microorganisms, bacteria are the most abundant, which means they are a lot, okay? In number, they are very high. Some of these bacteria are known to live in extreme habitats, such as extremely hot temperature or extremely low temperature, very acidic conditions, a place where there is no oxygen available. So in all such extreme habitats, bacteria are known to function. Many of them live on or inside other organisms is parasites. I'm sure you know by now as biology students what parasites are. Parasites are a type of heterotrophic organisms. They themselves cannot synthesize food. They will depend on another living organism for nourishment, and they will derive nourishment from this living host. Okay. Now, these can act as parasites on the surface of the body of other organisms, or they can live inside the body of other organisms. Okay. Now, we'll move on and talk about the shapes of bacteria. Bacteria come in many different shapes. Some of them are mentioned here. There could be spherical. If they are spherical, we call them cocci. Coccus is singular, cocci is plural. If they are rod shaped, it is called bacillus. If they are spiral, we call it spirillum singular, spirilla plural. Some of them are comma shaped. If they are comma shaped, we call them vibrio. I'm sure you've heard of Streptococcus, Staphylococcus. They all are spherical bacteria. Bacillus thuringiensis, it's a rod-shaped bacterium. Spirilla, Helicobacter pylori, it's helical. So, that is the example. Vibrio cholerae, the bacterium that causes cholera, that is an example for a comma-shaped bacterium. Okay, so bacteria can come in these different shapes. Now, let's talk about how different the mode of nutrition is in bacteria. If you think about it, structurally they are very, very simple. They're very simple organisms. They don't even have a well-defined nucleus. They don't have a lot of organelles within them. They are simple when it comes to structure. Even chemical composition, if you think, they're not very, very, complex. But behaviorally, in mode of nutrition, they are very, very diverse. They are very complex. Within this one group, remember when we spoke about fungi, I told you they are strictly heterotrophs. When I told you about plantae, I told you they are strictly autotrophs. Here, that is not the case. When it comes to how they derive their nourishment, there are so many different types. That means there is diversity, there is variety when it comes to mode of nutrition. So in NCERT, there is a line mentioned which says it shows the most extensive metabolic diversity. Many different types of mode of nutrition you can see within Monera kingdom. One type is, it can be autotrophic or it can be heterotrophic. Autotrophic means it has the ability to synthesize its food. Heterotrophic means it doesn't have that ability. It depends on other organisms. Now, while being autotrophic, it can obtain energy by synthesizing food by utilizing energy of the sun. If organisms are trapping the energy in the sun, performing photosynthesis, preparing glucose, we say they are photoautotrophic. They can take care of their energy needs, but for that they will utilize the energy from the sun. Within bacteria, there are some bacteria called cyanobacteria within Monera, which is capable of performing photosynthesis, so we can call them photoautotrophic. Just because an organism is autotrophic, capable of meeting its energy needs by itself, doesn't necessarily mean it should always depend on sunlight. It will always perform photosynthesis. There is another way in which organisms can be autotrophic. We call that chemoautotrophic. In chemoautotrophic, what happens? Going forward, I will explain to you. Basically, they obtain energy from oxidizing certain chemicals. Okay. Coming to heterotrophic bacteria. Heterotrophic means they themselves cannot synthesize, they depend on other organisms. One way is parasitic. They depend on another organism which is called the host, and that organism is living. Okay, it depends on a living host. They can be saprotrophic, which means they depend on another organism. They depend on organic matter present in another organism, but that organism is no more alive. So basically they depend on dead and decaying matter. It could be plant matter, animal matter. So it depends on organisms, organic matter, but those organisms are not alive as in the case of parasites. Then we have symbiotic relationship. In symbiotic relationship or mutualism, what happens is two organisms are dependent on each other. Bacteria depends on another organism. In turn, the bacteria will be dependent on this organism. They are like friends living together, helping each other out. Alright. So these are the different types of modes of nutrition you can see in Monera. Now, in Kingdom Monera, you can categorize two groups. One is called 𝐏 bacteria, the other one is called U bacteria. Now what is 𝐏 bacteria? Archaic means what in general English? Something that's very ancient, very old. So this group of bacteria is believed to have evolved at a time so early on during the evolution of life on earth, that when these bacteria evolved, when these bacteria thrived, the conditions on our earth were not similar to how it is today. Back when they evolved, the temperature probably was very high, or there was not sufficient oxygen, there was anoxygenic conditions, or the pH was very low, it was very acidic. These are organisms that have evolved at that time. Even today, they thrive and they live in such harsh, extreme climatic conditions itself, okay? They will not be able to survive under normal conditions like us, because they evolved at a time, once upon a time, very long ago, archaic, ancient time, when the conditions around itself were like that, okay? So even today, they are found in similar conditions. Under 𝐏 bacteria, we have three different types. One is thermoacidophiles, one is halophiles, and the other one, there are three of them. One is thermoacidophiles, the second one is halophiles, and the third one is called methanogens. What are thermoacidophiles? It's in the name itself, right? Thermo relates to heat or temperature. Acidic, acido. So these are organisms that are found in extremely high temperatures like 80 degrees Celsius, where we could just melt away. So in those kind of habitats, these organisms live. They live in hot water springs and those places where the temperature is extremely high, and also acidic, like very low pH. What are halophiles? Halo refers to not the halo around the head. It refers to salinity. Right? So halophiles, philes means philia, liking towards something. So these halophiles are those organisms that are found in extremely salty conditions. Okay? And methanogens. Methanogen, it's in the name itself. Gen means generating. Methano means methane. These are the organisms that have the ability to generate methane. How do they do that? They do so by anaerobically breaking down certain organic substances. And as a byproduct, methane is produced, and these organisms are used in the production of biogas as well, which we will learn later in the chapter Microbes in Human Welfare. For now, just remember, these methanogens generate methane. Where can we find them? We generally find them in the gut of ruminant animals like cow and buffaloes, like in cattle. And yeah, they are present in marshy areas. They are anaerobic in nature. They can survive without the presence of oxygen, and they produce biogas. Now you might ask me, how is it that these organisms are capable of surviving in such extreme habitats where not many organisms can survive, right? So the reason lies in the structure of its cell wall. The structure, it's very different from the cell wall of other bacteria. Because of the modified structure of its cell wall, it is capable of surviving even in such extreme harsh habitats. Okay, so archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme climatic conditions. Okay, now let's talk about eubacteria. In eubacteria, you have so many different types. Eu means true, prokaryotic, eukaryotic. Eukaryotic means true nucleus. Eu means true. So these are called true bacteria. There are several species of bacteria present, true bacteria. And these true bacteria do have a cell wall. And that cell wall is made up of a substance called peptidoglycan. Some of these bacteria are motile, which means they are capable of movement. If they are capable of movement, obviously they should have some locomotory structure that will help it move around. If they are motile, then they will have flagellum. Some of these eubacteria are autotrophic, some of them are heterotrophic. First, let's learn about autotrophic bacteria. Remember, I told you cyanobacteria are autotrophic, right? So we'll learn about cyanobacteria first. So these are photosynthetic, they are photoautotrophic. They perform photosynthesis very similar to how higher plants perform photosynthesis. They will trap the energy present in the sunlight, convert it into glucose and all of that. Okay. So these are also called blue-green algae. You know what cyan means, right? What color is cyan? Cyan is blue-green color. Cyanobacteria are called so because of its color. Okay, so these are also called blue-green algae. Initially, they were thought to be algae because they are photosynthetic, but now we call them cyanobacteria because they are prokaryotic. Okay, this is capable of photosynthesis. Obviously, if it can perform photosynthesis, one absolute necessity is the presence of pigments like chlorophyll A. So this also contains chlorophyll A. Some of these cyanobacteria are unicellular, some of them are colonial, some of them are filamentous. What you're looking at here, this is a diagram from NCERT. This is a diagram of nostoc. As you can see clearly, this is filamentous in nature. What do I mean when I say it's filamentous? I mean you have cells here lying one on top of another like this. And it forms a long thread-like structure, so we call it filamentous. Okay, so as you can see, this is example for a filamentous cyanobacteria. Where do you find them? You generally find them in aquatic habitats. Majority of them are found in freshwater habitats. Some of them are found in marine environments as well. Very rarely some terrestrial cyanobacteria also you can see on land, but they are found only in regions where it is moist. There is moisture. Okay. And these are covered by a gelatinous sheath. Surrounding this, there will be a gelatinous or a mucilaginous sheath, as you can see here. Okay, and another thing is, some of the cells in this filament, like the one that I am outlining in pink color right now, like this cell over here, if you notice, these cells look different from the other cells. These cells are called heterocysts. Okay, I'll tell you what the function of this heterocyst is. You know, plants require nitrogen. They generally absorb nitrogen from the soil. Plants cannot absorb nitrogen from air. Okay, so if nitrogen has to be converted from atmosphere to some form that plants can absorb, such as ammonia, nitrates, nitrites, the process by which atmospheric nitrogen is converted to ammonia or some other forms that is absorbable is called nitrogen fixation. If living organisms are involved in the process of nitrogen fixation, we call it biological nitrogen fixation. Some organisms perform biological nitrogen fixation. They convert the nitrogen that is present in the atmosphere, molecular nitrogen, into forms that are absorbable by plants. Now, this nostoc has that very special ability to, perform biological nitrogen fixation. Okay, so this biological nitrogen fixation happens in cyanobacteria like nostoc in these specialized cells called heterocysts. In every cell of nostoc, it's not possible. It's only possible in cells like the heterocyst. It is in these cells only that the enzyme that is required for nitrogen fixation is present. Okay, so in the heterocyst, nitrogen fixation will take place. And sometimes, since they live in aquatic habitats in water bodies, because they multiply very rapidly, they grow too fast and form water blooms, or you can call them algal blooms. What do? It will form like a surface of, on the surface it will form a layer, continuous layer, which can sometimes secrete toxins into the water body and kill or harm the fishes and animals that are present there. If there are any submerged plants, because of this layer that this algae is forming or cyanobacteria is forming, light may not penetrate inside. So with that, the plants that are lying inside, lower in the water body, they will not receive sufficient sunlight. So it can cause a couple of problems. Okay, so sometimes this can form water blooms or blooms in water. Now, this is what it looks like. See, it's blue-green in color. This is a bloom that is formed from cyanobacteria. I'm sure you're able to notice. Here it is clearer water. I want you to notice this portion where there is a film of cyanobacteria growing. That is called bloom. Okay. Now, that was about photosynthetic bacteria. Now let's talk about chemoautotrophic bacteria. They are autotrophic, capable of taking care of their energy demands by themselves by not depending on other organisms. How do? They do it by oxidizing certain chemicals. They oxidize various organic substances such as nitrates, nitrites, ammonia and all of that. And whatever energy is released during this oxidation, they will use that energy to synthesize ATP. You know how in nature all the nutrients, all the minerals will get cycled between living and non-living things, right? The nitrogen that is present in my body after I'm dead, it will go back to the soil. A plant will take it up, some animal will eat it. So it gets cycled through nature. Because of bacteria like these, chemosynthetic bacteria, cycling of many minerals will take place. Okay, so it recycles nutrients such as nitrogen, phosphorus, iron, sulfur, etc. So that is about autotrophic bacteria. Now let's move on to heterotrophic bacteria. Heterotrophic bacteria are organisms that depend on other organisms for food. Okay. If you compare autotrophic and heterotrophic bacteria's abundance, relative abundance, heterotrophic bacteria are much more in number compared to autotrophic bacteria. These are the most abundant in nature. Most of these are decomposers, which means it is saprophytic. They depend on dead and decaying matter. After a leaf falls off of a tree or some organism dies, it gets decomposed, right? Its body gets broken down. It gets eaten up by microbes. So this is one of the major microbes that is involved in breaking down of organic matter in dead organisms. Okay, so that is why we call it a decomposer. Some of these bacteria play a very important role in our lives. We might look down on them because they cause diseases, but some of these bacteria are really very helpful. We have to thank bacteria for curd and some other milk products like cheese, because fermentation happens, formation of curd happens because of certain bacteria. More about it we learn in the chapter Microbes in Human Welfare. For now, just know some bacteria are involved in the production of antibiotics. Some of them fix nitrogen. Remember, I just told you about nostoc and anabena, right? They fix nitrogen. So you have different types. Next, some of them cause diseases as well. Cholera, typhoid, tetanus, citrus canker in plants. All of these diseases are caused by pathogenic bacteria. So there's good bacteria and there are bad bacteria as well. Now, let's talk about how they reproduce. They reproduce by a process called fission. It is very simple type of cell division. It's not like mitosis where spindle apparatus is involved. Over here, it's a very simple type of cell division where the DNA, the genomic DNA that you have in bacteria, it makes an extra copy. It moves to the other side of the cell. A cell wall forms in between and the two cells are formed. This is called binary fission. Fission means what? Splitting. Binary, two. One cell is splitting to give rise to two cells. Very simple kind of binary fission it will undergo. Generally, this happens when the conditions are favorable. Sometimes when the conditions are not favorable, when they are unfavorable, it will produce certain types of spores. As you can see in these micrographs, images taken from under the microscope, you can see these circles here, right, with very thick walls. These are the spores. When conditions are unfavorable, it will form spores to tide over the unfavorable condition. Now, both of these are asexual reproduction. Talking about sexual reproduction in bacteria. Generally, when we talk about sexual reproduction, people tend to think about how gametes are formed and gametes are going to fuse, form a zygote. All of that story and drama is not going to happen in bacteria. It's a very primitive type of sexual reproduction. Just exchange of genetic material itself is considered as sexual reproduction. Some bacteria have a structure called pilus. More about it we'll learn later. It will form like a tube, conjugation tube between two bacteria. This bacteria's genetic material makes an extra copy and it gets passed on. Like it will have a DNA, a type of DNA called plasmid, and that will get passed on through this tube to another bacterium. That itself is considered sexual reproduction. It's a very primitive type of sexual reproduction which does not involve either formation or fusion of gametes. Okay, just DNA is getting transferred, that's all. Now, mycoplasma is also included under Monera, but mycoplasma is very different from other bacteria, both eubacteria and cyanobacteria, because this mycoplasma does not have cell wall at all. It lacks cell wall. But why are we including it under Monera? Because it is prokaryotic. Around its DNA, there's no well-defined nuclear membrane. Okay, that's the reason it's grouped under Monera. Now, we know bacteria are small, they're super tiny. Compared to eukaryotes, very small. But among bacteria, the smallest type of organism is mycoplasma. Okay, so it is the smallest living cell known. These also have the ability to survive without oxygen. Many of these mycoplasmas are pathogenic to plants and animals, which means they can cause disease in plants and animals. Alright, so that's about Monera. What all did we learn in Monera? In Monera, we learned about the shapes of bacteria. We learned about how there is metabolic diversity. We started with autotrophic bacteria, first photosynthetic or photoautotrophic like cyanobacteria, then chemoautotrophic bacteria, then heterotrophic bacteria, how reproduction happens, just that. Okay, that's all you need to know about Monera. Now let's move on and learn about Protista. What do we already know about Protista? In Protista, we have eukaryotic organisms and they are unicellular. So we know that Protista includes unicellular eukaryotic organisms. In Protista, you have Chlamydomonas and Chlorella. Chlamydomonas and Chlorella, because they are photosynthetic, because they have a cell wall, they were earlier placed under algae in plant kingdom. But now that we know it is unicellular and eukaryotic, it's been moved away from it and included under kingdom Protista. Also, Paramecium and amoeba, which was earlier part of animal kingdom because they did not have cell wall. Remember, in Linnaeus' system of classification, plant kingdom and animal kingdom categorization depended on just one criterion, whether it has a cell wall or not. Because it had a cell wall, Chlamydomonas and Chlorella were earlier placed in plant kingdom. Amoeba and Paramecium and other protozoans, they were placed in animal kingdom. But now, since in the five kingdom classification, all the unicellular eukaryotic organisms are placed under Protista. These have been moved out of their earlier kingdoms and included under Protista. Okay, now the boundaries of these kingdom are not very well defined. If you talk about fungi or plantae or animalia, the other eukaryotic kingdoms, you know for sure what is the mode of nutrition in all of them, right? But here that is not the case. You have some organisms that are like plants and photosynthetic. Chrysophytes, euglenoids, dinoflagellates, all of these are photosynthetic like plants. So, since they are capable of performing photosynthesis, let's say they are plant-like. Slime molds are like fungi with respect to their mode of nutrition. I mean, they are saprophytic. Talking about protozoans, which includes Paramecium and amoeba, these are animal-like because they show holozoic nutrition or ingestive heterotrophy. So, they are very diverse, very different kinds. We learn about each of them starting with chrysophytes. Chrysophytes, kingdom Protista. Okay, many organisms in kingdom Protista, they are aquatic in nature. They live in water habitats and they have a connecting link. They form a connecting link between plants, animals and fungi. I told you why, right? So, because we believe that the advanced plants and higher plants and animals would have evolved from these guys. Okay, so it forms a link. Some of them are locomotory, so they will possess cilia or flagella for locomotion. They undergo reproduction both asexually and sexually. Here, sexual reproduction is slightly more advanced than Monera. Over there, we just saw DNA transfer, but here actual gamete formation, gamete fusion, zygote formation, all of those events will take place. Okay. Now, take a look at this picture. It might look like one Diwali light arrangement, right? It looks like some mandala art, but actually these are a kind of protists that we call diatoms. Every cell that you're looking at here, like these, each one of these, and every one of these are a type of photosynthetic protists that we call diatoms. These are available in aquatic habitats. There's one person called 𝐄𝐍𝐆𝐋𝐈𝐒𝐇. He collects diatoms from different habitats, from ditches, from ponds and rivers and different places. He brings them home. He, arranges and makes artwork out of them. I want you to understand how special this is, because this is not something that you can do with your naked eye. All of this has to be done under the microscope because they are unicellular organisms which you cannot see with your naked eye. So it's a lot of work and it's beautiful. I just want to show this to you, and yeah. So these diatoms belong to a group called chrysophytes. Now chrysophytes along with diatoms also includes desmids. Desmids have a common name. They are called golden algae. Blue-green algae is cyanobacteria. Golden algae or golden brown algae is desmids. Since they are belonging to Protista, obviously they are unicellular and eukaryotic. Where can you find them? You can find them in fresh water as well as in marine environments. They are very small, they are unicellular. So obviously they will be microscopic. You will not be able to see them with your naked eye. And they are planktonic. What does that mean? They do not have any locomotory structures of themselves. They do not have cilia or flagella that help them move in water. If at all they have to move in water, if water moves, along with water, they will move or float passively. Okay. So, they are found to be floating on the surface of water. Because they float on the surface of water, we call them planktons. You may have heard of terms like phytoplanktons and zooplanktons. Planktons are basically organisms that are, floating on the surface of water. If they are settled at the bottom of the water body, we call them benthic organisms. These are planktonic because they're floating on the surface of water. Since this is photosynthetic, plant-like, we call this phytoplanktons. Okay, so most of these are photosynthetic, as you can see here. Now let's talk about the structure of these diatoms. The cell walls of diatoms are very special. Okay, like normally in plant cells, how you find a cellulosic cell wall, even over here cellulosic cell wall is present, but that cellulosic cell wall is embedded with silica. Because of the presence of silica, the cell wall is kind of indestructible. Now, normally you will find one continuous layer of cell wall, right? Here that is not the case. The cell wall is like a soap box. There are two parts to the cell wall. One part fits inside the other part like a soap box. So we say there are two shells that overlap. Each one can be called as a shell, and the two will overlap like that in diatoms. Okay, so the cell walls form two thin overlapping shells. You can see it here. This is how it will look. This is one part of the cell wall. Below that, having a smaller diameter, you have the other part of the cell wall, the other shell. They both will fit like a soap box. Okay, now there's something called diatomaceous earth that you have to know about. What I already said is the cell wall is pretty much indestructible, which means it cannot be destroyed. The reason is because it has silica in it. Okay, because of the presence of silica, because silica is embedded in the cell wall, it is not destructible. What happens after the organism dies? I also told you that these float on the surface of water. For as long as they are alive, there's metabolic activity, they will keep floating on the surface of water. But once these organisms die, what happens? The insides will decompose, but the cell wall will not decompose because it has silica. It will drop on the at the bottom of the water body. It will keep accumulating and form a sediment. In fact, it's accumulated over several thousands of years. It has been accumulating and it forms a sediment at the floor of the water body, which we call diatomaceous earth. So these cell walls, I said thousands, it is billions of years, it has gotten accumulated, and it forms a sediment that is called the diatomaceous earth. This diatomaceous earth has many commercial uses. It can be used in polishing, filtration of oils and syrups. Okay, and these diatoms are called the chief producers in the oceans. In the ocean, if you see, there are not many big plants, right? Majority of photosynthesis is performed by these phytoplanktons itself, like tiny little algae-like organisms. So they are responsible for performing most of the photosynthesis in oceans. That's why they're called as chief producers in the oceans. If you talk about terrestrial habitats, we have many big trees, but in oceans, that is not the case, right? So in oceans, they are called the chief producers. That's about your diatoms. About desmids, not much is mentioned. You just have to know that desmids also belong to chrysophytes along with diatoms. We have to know that desmids are also called golden algae or golden brown algae. Now let's talk about the second category of protists, which we call dinoflagellates. Dinoflagellates are photosynthetic protists that have two flagella. Okay, flagellate is having flagella, dino, two are there. In each dinoflagellate, there will be two flagella. Over here also the cell wall is made up of cellulose, but it is not like you see in plant cells. Cell wall is present in the form of stiff cellulosic plates. Okay. Though these are photosynthetic, they will have the green pigment chlorophyll. Along with chlorophyll, they will have many different pigments. Depending upon the abundance of which pigment, depending upon which pigment they have more abundantly, they can appear in different colors like yellow, green, brown, blue or red. It depends on which pigment is present in abundance in these cells. If there's more red color producing, imparting pigment, then it will appear red color. If there's more of brown color imparting pigment, then they will appear brown. So it depends on the relative abundance of different types of pigments. Okay. So yeah, most of these are marine, and like I already mentioned, these are photosynthetic. They are plant-like protists. Okay. Now, like I already mentioned, they have two flagella. How are they arranged? One of them is arranged longitudinally, one of them is arranged transversely. Transversely means horizontal plane. Longitudinally means vertical plane. Okay. See here, this is one dinoflagellate. You can see how one flagellum is horizontal, transverse, it's going behind like that and coming from here. So it's transverse. The other one is longitudinal like this. Even this one, you can see how this is transverse and this is longitudinal. I want you to also notice how cellulose is present in the form of plates here, stiff plates here, unlike other photosynthetic cells. Okay? That is why, since it appears like an armor, they are also called armored dinoflagellates. Some of these dinoflagellates, such as 𝐿, they are known to cause something called red tides. Red tides are basically like algal blooms that I told you about with respect to cyanobacteria. They multiply at a very rapid rate. They keep dividing, so much so that they kind of cover the surface of the water body. Since 𝐿 is a dinoflagellate that appears red in color, the water body surface will also appear red in color. We call that the red tide. This can sometimes release toxins into the water body, which will not show any change in color. It will not show any change in text, like how it appears. But if any cattle consumes them, if the fishes are exposed to it, fishes living in water, or if humans consume them, it can very badly affect their health. Okay, so it can even kill a lot of fishes that are present in that water body. Yeah, that's about dinoflagellates. Next we have bioluminescence to talk about. Bioluminescence means living organisms capable of emitting light. You may have seen fireflies, how they emit light. Some jellyfishes emit light. They are bioluminescent. Likewise, these dinoflagellates also are bioluminescent. Whenever they are disturbed, they are living in water, right? They live in water bodies. They are aquatic, and as a defense mechanism, they emit light. If there is some small animal like a zooplankton that is trying to feed on this phytoplankton like dinoflagellates, then to distract that organism, it will emit light. So whenever it is disturbed, it will emit light to scare off the predator. Okay, so that is called bioluminescence. Yeah, that's about dinoflagellates. Now let's talk about euglenoids. Euglenoids example is euglena. For a very long period of time, you may have learnt in school that euglenoids or euglena is a protozoan, but here it is changing. You're learning it under photosynthetic protists, under euglenoids, okay. So most of these are found in fresh water, still water, not moving water, stagnant water. They don't have a cell wall. Remember, dinoflagellates, diatoms had cell wall made up of cellulose. It was embedded with silica. Dinoflagellate had cell wall made up of cellulose, but it was in the form of stiff cellulosic plates. Here, cell wall is not there. Cellulosic cell wall is not there. Instead, you know, cellulose is a polysaccharide. It's a carbohydrate, right? Here, they have an outer covering called the pellicle, which is not made up of carbohydrate. It is made up of protein, okay? So, they have a protein-rich layer that is called the pellicle. Unlike the cell wall, which is very rigid, because of the presence of pellicle, the body of amoeba has a little bit of flexibility, okay. These also have two flagella, one very short that you won't even see coming out of here, and the other one is long. It has two flagella, okay. Now, I already mentioned earlier that these are plant-like protists, so we know that these are photosynthetic, but the problem is, they are not always photosynthetic. I've told you earlier how this is sometimes photosynthetic and sometimes heterotrophic. Similar to how higher plants have pigments like chlorophyll A and all of that, these also will have pigments. This is eukaryotic, it has membrane-bound organelle like chloroplast. All of that apparatus is present inside the cell. But for photosynthesis, light is extremely important. Higher plants, if they don't get sufficient sunlight, they die. But here, that is not the case. Whenever there is no sunlight available or light available to perform photosynthesis, this will turn into a heterotroph. It will engulf smaller organisms, feed on it and act like an animal. Okay. So this is, whether this is autotrophic or heterotrophic depends on the availability of light. Okay. So they are photosynthetic in the presence of sunlight, and when deprived, presence of sunlight. Yeah. And when they are deprived of sunlight, they behave like heterotrophs. Okay, by eating other smaller organisms. Since they are photosynthetic, obviously they should have pigments. They have pigments that are very similar to the pigments that we find in higher plants. Okay. Now, we'll talk about the slime molds. I told you that slime molds are fungi-like protists, which means that they are saprotrophic. Okay. So, in slime molds, you have two different phases. In one phase, called the plasmodium, what happens is, whenever the conditions are favorable, these small single-celled eukaryotic cells, they are very similar to amoeba. It's like many small cells like this. They will come together, they will fuse. Many such cells will come together, like many drops of water are coming together. Many of these cells will come fuse and form a structure called the plasmodium. Okay, this is called the plasmodium. This plasmodium will keep on dividing, increasing the number of nuclei. It will start moving on dead and decaying organic matter like twigs and old leaves and all of that. It will start, getting the nourishment from those dead and decaying matter, similar to how fungi do. And whenever conditions are not favorable, this plasmodium will transform into a fruiting body. Within this fruiting body, spores are produced, and those spores will be released. And those spores will tide over unfavorable conditions. Okay. So, these are saprophytic protists. The body, which we call the plasmodium, it will move on decaying twigs and leaves, obtain nourishment from it, absorb from it. Whenever suitable conditions are there, they will form the plasmodium. And this plasmodium may spread across several feet in the forest, as you can see here. Okay, all of this is plasmodium. But when conditions are not favorable, the plasmodium will differentiate and form fruiting bodies like this. Within the fruiting bodies, spores will be produced. The plasmodium does not have cell wall. In this, there is no cell wall. But In the spores that are produced, there will be cell wall because cell wall is protective in nature. The conditions as it is are not favorable. If you have to tide over unfavorable conditions, you need a protection, right? So that's why when spores are formed, the spores will have a cell wall, and that cell wall is made up of cellulose. Okay? And these cellulosic thick-walled spores, they are very resistant to harsh climatic conditions. They can survive for several years, and whenever conditions are favorable, whenever favorable conditions return, the spores will germinate to give rise to the plasmodium itself. So these spores are germinated to far off places. So probably if the conditions are better there, then these spores can germinate. Okay. Now let's talk about the last type of protists, which is protozoans. Protozoans are animal-like protists, which means they are all heterotrophic in nature. They either kill other organisms and eat them, or they will parasitize other organisms, depend on other live host. Okay, so they can be predators or parasites. They are believed to be ancient relatives of animals. It is believed that we evolved from these protozoans. Depending upon the locomotory structure of these protozoans, we can categorize them into 4 types. You have amoeboid protozoan, flagellated protozoan, ciliated protozoan, and sporozoan. Amoeboid protozoan, example is entamoeba or amoeba you can take, right? In amoeba, you know, the locomotory structure is called pseudopodia. They contain pseudopodia. They are generally found in fresh water or marine water and on terrestrial habitats if there is moisture. In moist terrestrial habitats you can find them. These are called pseudopodia. You know pseudopodia, right? It can change its shape. So this amoeba moves around with the help of pseudopodia. Marine forms, some of these marine forms will have silica shells on their surface for protection. Some of them are pathogenic. You know, entamoeba histolytica causes amoebic dysentery, right? That belongs to amoeboid protozoan itself. Now, let's talk about flagellated protozoan. Example is trypanosoma. Earlier, you may have learnt of euglena as an example for flagellated protozoan, but we've already covered that under euglenoids. So here the example that you have is trypanosoma. In order to move around, it uses flagella as the locomotory structure. They can either be free living or they can parasitize other hosts. Some of these parasitic forms cause diseases. For example, sleeping sickness in humans is caused by trypanosoma, which is a flagellated protozoan. Now let's talk about the second last type, ciliated protozoan. In ciliated protozoan, obviously as the name itself indicates, you will have cilia present. Cilia present all across the surface of these organisms, and they help to move food into the body of the organism. They help the organism also move around. Similar to how in Kerala boat race you see many men are sitting and they're all at once rowing the oars in the boat. Very similarly, this also works like the oars of a boat. They all beat in one direction and the organism will move forward. Okay, so there is a cavity that this organism has called the gullet. Into this food has to go. So the beating of cilia will push food inside the gullet. Okay, and yeah, that is what allows them to move. Example for that is paramecium. Okay, that's about protozoans, ciliated protozoans. Now let's talk about sporozoans. Sporozoans have no locomotory structures. They are called sporozoans because in their life cycle, there will be an infectious spore-like stage. Example for this is plasmodium. The plasmodium that we learnt in the life cycle of slime molds is a phase in the life cycle. It's not the name of an organism. But in protozoans, plasmodium is the name of a species of protozoan. It is the species that is known to cause malaria. I'm sure you've learnt about Plasmodium vivax, right? So it is the same Plasmodium here. You know, Plasmodium vivax is also causes infections. It has a spore-like stage, right? So that is why it's included under sporozoans. It causes malaria. Yeah. And that's all about protozoans and protists. What we have next on our list is kingdom fungi. Fungi, as we already know, are heterotrophic organisms. They cannot synthesize their own food. They depend on other organisms for food. They show very high diversity when it comes to their habitat as well as morphology. Habitat means the place where they are found. Okay. Now, how are they distributed? They are cosmopolitan in distribution, which means they are found pretty much everywhere on earth. On all different types of habitats, you can find them. You can find them on land, you can find them in the air, you can find them in water. So they're pretty much found everywhere. That's why we say they are cosmopolitan in distribution. That means they are everywhere. Okay. But generally, when it comes to the climatic conditions, they prefer to grow in areas that are warm, not too hot, not too cold, just the right amount of warm, and also a little humid or moist. That is the best place for fungi to grow, which is why you may have seen during monsoons when it is humid and when it is a little warm, these fungi grow very well. And the reason we refrigerate food is because at extremely low temperatures, fungi do not grow. So that's why, to save the food for a longer period of time, to increase the shelf life of food, we store them at very low temperatures in the refrigerators. Okay. Now, we already know that fungi have cell wall. The cell wall in fungi is made up of a substance called chitin, which is a polysaccharide. Along with chitin, there could be other polysaccharides as well. Now, let's talk about how we are aware of fungi. I'm sure everybody at some point in your life would have seen some mold growing on bread. You may have seen a fruit rotting. So all of these greenish white cottony patchy kind of structures that you see on bread, on fruits, they're basically fungi growing. Okay. And the mushrooms that you eat, these beautiful pretty toadstools that you see on Nat Geo and Animal Planet, they show these, right? So these are called toadstools. The reason is, as you can see here, this resembles the mushroom in having an umbrella-like shape, and toads often like to sit on top of it and use it like a stool. That's why they're called toadstools. Though both of these are fungi, generally mushrooms are edible. Many mushroom species are edible, but toadstools many of them are not edible. Okay. Now, yeah, we all know about yeast. Yeast is a unicellular fungus. It is used in the brewing and baking industry, Saccharomyces cerevisiae. More about it we'll learn in the chapter Microbes in Human Welfare. So yeast is used in the baking industry to prepare breads and cakes and all of that. And in the brewing industry, it's used in the preparation of alcoholic beverages. Okay, so that also is a fungus. Penicillium is a fungus that is used to obtain an antibiotic called penicillin. More about this we will learn in the chapter Microbes in Human Welfare. So, fungi have many uses, and it also is known to cause some diseases in plants as well as animals. This is a mustard plant leaf. On the mustard plant leaf, you can see these white small spots. This is called the white rust disease of mustard, and it is caused by a fungus called albugo. And here you can see this is the stem of wheat. On the stem of wheat, you can see rust colored patches. So this is called the wheat rust disease, and this is caused by a fungus called Puccinia. Generally, animal diseases are majorly caused by bacteria, while plant diseases are majorly caused by fungi. Similar to how we have beneficial bacteria and harmful bacteria, you have fungi which are useful and fungi that cause diseases in plants and animals. Okay. Now, generally, in most cases, fungi are multicellular. However, there is an exception. Yeast is a unicellular fungus. Every time yeast is mentioned, people think about Saccharomyces cerevisiae, but that's not the only yeast species. There is another species of yeast called Monascus purpureus, which you will learn about in the chapter Microbes in Human Welfare. So, yeah. There are exceptions, and yeast is an exception where it is unicellular but still grouped under fungi, because under certain conditions it forms a fungus like hypha, which is called a pseudomycelium. So that is one of the reasons why it is grouped under fungi. It has a chitinous cell wall also like fungi. Okay, so All other fungi except for yeast, they are multicellular. Okay. Now let's talk about the structure or the morphology of fungi. It's very simple. Whenever it is multicellular, the cells are like this, filamentous in nature. They lie one on top of another, form thread-like filamentous structure. We call these the hyphae. Singular hypha, plural hyphae. Those thread-like structures are called hyphae. Over here, see, do you see this thread-like structure here? This is hyphae. This hyphae, when it forms a network, we call that the mycelium. Okay, what you see here is the mycelium. You take a portion of this, look at it under the microscope, here is what it will appear like. You can see hyphae here. The network of hyphae is what we call mycelium. Okay, now, yeah, this is what the mycelium will look like. These hyphae, since they are multicellular, many cells are present. In between two adjacent cells, a cross wall or a septa may or may not be present. In some species, in the hyphae, between the cells, between two neighboring cells, cross walls will be present. If cross walls are present, the cross walls are also called septa. So whenever cross walls are present, such a hyphae or mycelium is said to be septate, which means septa is present. Okay, I want you to see here how this is one cell, this is one cell. In between the cells, you can find the presence of septa, right? So septa are present. So we call it septate. Here is an actual micrograph of a septate mycelium. Notice how over here you can see there are cross walls present clearly between cells. Right? So whenever cross walls are present, we say that they are septate. In contrast, in some species, between cells, cross walls or septa will not be present. When septa is not present between two cells, what is going to happen? It will appear like a continuous tube that is filled with cytoplasm and have many nuclei. Alright, so in such a case, we call it an aseptate mycelium or cenocytic hyphae. Okay, some of them are continuous tubes filled with multinucleated cytoplasm. You call it cenocytic. See here. Since there are no cross walls or septa between, it will appear like a tube with cytoplasm. It has many nuclei, of course, but between these nuclei there are no cross walls. So it is multinucleate condition. We say it is cenocytic. This is the correct spelling. Okay. So here is a micrograph of a cenocytic hypha where you do not see any cross walls present in the hypha. Okay. Now, here is a comparison between septate and aseptate or cenocytic hypha. This is also called aseptate, which means septa are absent. Okay. So here cross walls are clearly visible. See, over here it is not visible, it's not present. This is considered to be an advanced feature. This is considered to be a primitive feature of fungi. Now let's talk about how, what are the different modes of nutrition you can see in fungi. Of course, they are all heterotrophic. And within heterotrophy, what different types of modes of nutrition can you see? Some of them are saprophytic, not some of them, majority of them are saprophytic. Some of them are parasitic, some of them are symbiotic. Saprophytic means, I've already mentioned this earlier, they depend on other organisms that are dead and decaying. So it will secrete enzymes outside, absorb the nourishment from whatever is present around it. Okay, so it absorbs soluble organic matter from dead substrates. These are important because they break down organic matter and help in its recycling. Parasitic means they depend on another organism that is living. It will parasitize it and derive nourishment from that organism. Sometimes because it is a parasite, it will cause diseases in plants and animals. So it absorbs nourishment from a living host. It causes harm to the host. It could sometimes even kill the host. Okay. Symbiotic, I've already told you about this. Symbiotic is a mutualistic relationship where two organisms are involved and both of them benefit because of that interaction from each other. Okay, so it is a mutualistic association. Example, we have lichens and mycorrhiza. In lichens, fungi and algae, they are closely associated. Fungus helps in absorption of water and minerals. It helps in providing shelter to the alga. Alga being photosynthetic, in return will synthesize food and provide to the fungus. Fungus helps in absorption. In return, alga gives it food. Mycorrhiza. Mycorrhiza are symbiotic associations that fungi have with the roots of higher plants like gymnosperms and angiosperms. What basically happens here is similar to how it helps algae in lichens, it helps roots of higher plants in increasing the efficiency of water and mineral absorption, and also in return the plants will provide food to the fungus. Okay, so that is lichens and mycorrhiza. Now let's talk about how reproduction occurs. In fungi, we can see vegetative reproduction, asexual reproduction as well as sexual reproduction. How does vegetative reproduction happen? What is vegetative reproduction? Vegetative reproduction is when reproduction is happening because of some somatic cells in the hyphae. If you think about how vegetative propagation or reproduction happens in plants, it doesn't happen because of flowers, it happens because of the vegetative parts like the root, stem and leaves. You take a rose stem cutting and plant it in your house, it becomes a new plant. So if you're using vegetative parts, it becomes vegetative reproduction. Here, there is no root, stem and leaf. Here, the somatic cells of the hyphae, if that is used, in reproduction, it is called vegetative reproduction. In vegetative reproduction, you have three types. You have fragmentation, fission and budding. We learn about it in detail. And in asexual reproduction, specialized structures are formed. Within these specialized structures, or with the help of those specialized structures, spores will form. There are three different types of spores: sporangiospores, conidiospores and zoospores. We learn about them as well. Sexual reproduction happens by means of spores again, but these are sexual spores. Example for them are oospores, ascospores and basidiospores. More about it in detail we learn. The difference between how these spores are formed and how these spores are formed is, in asexual reproduction, spore formation happens by mitosis. In sexual reproduction, spore formation happens because of meiosis. Understood? Now, in fungi, we see that spores are formed in distinct structures that we call fruiting bodies. I told you about mushrooms, right? I showed you the picture of mushrooms. The mushrooms that you eat, the part of the fungus, the mushroom that you eat is actually a fruiting body. So many different types of fruiting bodies form, and with the help of those fruiting bodies, spores are released. Okay? Now, let's talk about vegetative reproduction first. We have three types: fission, budding and fusion. Fission and budding are seen in unicellular fungi. Fragmentation is observed in multicellular fungi. What happens in fission is very similar to what happens during binary fission in bacteria, but over there it's not mitosis. Over here, proper mitosis will happen. It's a eukaryotic cell. So this is a species of yeast called budding yeast. So here you can see the cell will initially be small. It will become bigger, elongate, duplicate its genetic material, make an extra copy of DNA, and then split to give rise to two cells. From one cell, you always get two cells. See, this used to be one cell, it is now two cells. This is now in the process of becoming two cells. So here, fission is happening, splitting is happening, right? By undergoing mitotic cell division, one cell is giving rise to two cells. That's why it's called fission. Now, what is budding? Budding is very similar to fission because that is also a vegetative reproduction. But in budding, from one cell you can get you can have more than one bud. Okay. So I want you to notice here how this is one cell. This one cell forms small outgrowths like this, as you can see here, like teddy ears. There could be more than one such small outgrowths, which we refer to as buds. DNA replication will happen. DNA is moved here and that bud will eventually get pinched off and become a new organism. You can see this in budding yeast. Okay. Next we have fragmentation. Like I already mentioned, fragmentation happens when in the fungal species there are multicellular. So in multicellular fungi you have hyphae. In hyphae you have many cells that are it is joined to each other. Now in this, let's say one of the cells in between gets damaged or dies, then what happens? This one hypha is now split into two hyphae. Each one will act like a new organism. What does fragment mean? Fragment means pieces, right? Over here, one single hypha that was there is broken into smaller fragments for whatever reason. A cell could die in between, or it could get damaged, or something happens. So whenever smaller fragments are formed, each fragment is equivalent to a new organism. So that is called fragmentation. Now let's talk about asexual reproduction. Asexual reproduction, you have three different types of spores: conidiospores, sporangiospores, and then we have zoospores. Okay, in all of these three spores you should remember two things: whether it is produced inside a structure or it is arising from outside of a structure. If it is produced inside a specialized structure, we call it endogenous. Endo means inside. Endogenous means it's getting generated inside that structure. Exogenous means outside. That is one thing that we should remember. And another thing is whether it is motile or non-motile. Does it have any locomotory structures like flagella or not? Okay. First, let's talk about conidiospores or conidia. Conidia do not have any locomotory structure, so they are non-motile. They are produced on top of the conidiophores. So they are not produced inside any structure, specialized structure. So you can say they are exogenous. They are non-motile and exogenous. Exo means outside. What basically happens is hyphae, usually they are found below the substratum. They are found to grow on top of the substratum, right? I'll show it to you here. So the hyphae will be growing, let's say, under the soil like that. If it has to produce conidia, then it will give out branches aerially. Okay, so branches will start coming out on the surface aerially. From the soil it will start appearing outside. These aerial branches will bear structures called conidiophores. F-O-U-R, not F-O-U-R, not F-O-R-E, it is P-H-O-R-E, conidiophore. Phore means, I'll write it down here, conidiophore. Phore means bearing. It means it's bearing something. Conidiophore means a stalk-like structure that is bearing conidia. Okay. So at the tip of this conidiophore, these conidia are present. And each of these rounded conidia or conidiospores will get pinched off and get released as spores. Since these are not formed inside specialized structures, they are being released from the tip of the conidiophore. They are called exogenous. Okay, when these spores germinate, you get a new hypha. Okay, see, this is present below the surface. One branch will arise aerially. It will bear structures called conidiophores. Conidiophores at their tip will bear conidia. Conidia are also called conidiospores. Don't get confused between conidiophore and conidiospore. Conidia are called conidiospores. Conidiophore is the stalk that bears the conidia. This will get pinched off from the tip and those spores are released. Once these spores germinate, it will give rise to a new hypha. That's about conidia. Next we have sporangiospores. These are also called aplanospores. These also do not have any locomotory structure, so they are non-motile, but they are produced inside sporangia, structures called sporangia. So they are endogenous. They are non-motile, but they are endogenous. I want you to notice here, this is a hyphal branch. Here you can see at the tip there is a structure called the sporangia, or the sporangium is singular. Within this sporangium, spores are present. Those spores are called sporangiospores. These are non-motile spores. When these when the sporangium breaks open and releases the sporangiospores on finding a suitable substratum will germinate and give rise to new hyphae. Okay, the difference between that and this is that is exogenous, this is endogenous. Okay, now we'll talk about the last type of asexual spore, which is zoospores. Zoo means motile, right? Zoospores obviously are motile because of the presence of flagella. Generally, each zoospore will have 2 flagella and these are usually found in aquatic fungal species. They are also produced inside specialized structures called zoosporangia. Over here you can see this is a zoosporangium. Within the zoosporangium, these zoospores are formed. Each zoospore has 2 flagella that will help it move within the water body. Okay, that's about asexual reproduction. Now let's move on and talk about sexual reproduction. Now in fungal sexual reproduction, there are 3 very important steps. We'll talk about that. But before that, understand, unlike majority of plants and animals who are diploid, like we have 2 sets of chromosomes, one that we receive from the mother, one that we receive from the father. Majority of fungi are haploid in nature, which means in the hyphae, in each cell, you will find only 1 set of chromosomes in the nucleus. Okay, so they are haploid. So the first thing that happens is 2 cells, when they are fusing, 2 cells of opposite mating types we call. When the 2 cells fuse, first the protoplast will fuse. The protoplasm of these 2 cells will come together and fuse. That process is called plasmogamy. Let's start from here. Here we have the mycelium, right? This is the mycelium. Let's say there are this 2 mycelia here of 2 opposite mating types. Let's call this plus, let's call this minus. Both of them belong to the same species, but they are of opposite mating types. Similar to how we have males and females, here you call it opposite mating types. Let us differentiate this by using different colored nuclei. Okay, so one of this we'll make pink, the other one we'll make blue. So these cells are haploid. Let's say this cell right here and this cell, they will fuse. 2 cells of opposite mating types, they fuse. The process is called plasmogamy. When they first fusing, only the protoplasm will fuse. The fusion of protoplasm is called plasmogamy. What is a consequence of plasmogamy? Within the same cell, the protoplasm would have fused, but the nuclei will be separate. Within the same cell, you will have 2 nuclei, one pink, one blue, 2 opposite mating types nuclei will be present, not necessarily pink and blue. I'm just telling you because I've used pink and blue here for reference, just so you understand. Okay, so you will have one pink and one blue, which indicate they are from 2 opposite mating types. Okay, so this phase is called dikaryotic stage, or this is called a dikaryon. It contains 2 nuclei. You know, karyon means nucleus, di means 2, right? So within the same cell, following plasmogamy, you will have 2 nuclei. Eventually, these 2 nuclei will also fuse to form the zygote. Okay, so the fusion of nuclei is called karyogamy. Gamy means coming together of fusion. Karyon fusing is karyogamy. But now, in many advanced species of fungi, advanced groups of fungi, it will remain in the dikaryotic stage for a considerably long period of time, and eventually later only it will form the zygote. It can remain in this phase for a long period of time. Okay, so this is called the dikaryotic stage or dikaryophase. What we need to understand here is, though plasmogamy has happened and within the same cell you have 2 nuclei, this does not make it diploid. 2 nuclei have not yet fused, they are separate. It is haploid, but it is multinucleate. Basically, it has 2 nuclei. Both the nuclei are haploid. Dikaryotic phase is not diploid. Okay, so it will stay this way for a while. The first step here is plasmogamy. As a result of plasmogamy, dikaryotic stage is formed. The second step is karyogamy. It may or may not happen immediately depending upon the species. So once the nuclei fuse, you will have a diploid zygote forming. As a consequence or product of karyogamy, the zygote will form. In the zygote, which is diploid, meiosis or reductional cell division will take place. As a result of that, haploid spores are formed. Now, these spores will germinate and give rise to the mycelium again. Understood? So, first step, what happens? Plasmogamy happens. Plasmogamy is the fusion of the protoplasm of the two cells, which may or may not be, which may be motile or non-motile. As a result of plasmogamy, we get a dikaryotic stage. Eventually, the two nuclei will fuse by a process called karyogamy. As a result of karyogamy, the zygote will form, which is diploid. The zygote will undergo reductional division or meiosis and give rise to haploid spores. The spores will germinate to give rise to new mycelia. So, this is how sexual reproduction happens, and while sexual spores are being produced, meiosis will take place. This is the third step. Okay. Now, let's understand the classification, further classification of fungi. Fungi are further classified based on the morphology of mycelium and also how sexual reproduction takes place. Remember when we spoke about Whitaker's 5-kingdom classification, I told you we consider a couple of criteria. Over there I mentioned reproduction as well. Here is an example of how reproduction can be used as a criterion for further classification of kingdoms. Okay. So in kingdom fungi, further classification, first thing that we are going to look at is its mycelium or hyphal structure, whether it is septate or aseptate. I've already told you earlier, aseptate or cenocytic condition is primitive, septate is advanced. If it is aseptate or cenocytic, then it belongs to a group of fungi called phycomycetes. Phycomycetes are also called algal fungi. Actually, under phycomycetes, you have two other groups, which is zygomycetes and oomycetes, but this differentiation has not been done in NCERT. Now, under septate mycelium, based on what kind of spores are formed, what kind of fruiting bodies are formed, you further classify this into three groups. They are ascomycetes, basidiomycetes and deuteromycetes. Ascomycetes are called sac fungi. Basidiomycetes are called club fungi. Deuteromycetes are called imperfect fungi. Okay. Now, let's start with phycomycetes. Examples for that, it's on the board: mucor, rhizopus, albugo. Remember, I told you about albugo. It causes rust disease on mustard leaves. Remember, I told you white rust. It's the same thing. Habitat, where do you find them? Some of these are aquatic. Okay, and they can also be found on decaying wood, moist and damp places. Some of them are obligate parasites on plants, which means they 100% mandatorily require a host plant. Without the host, it cannot survive at all. So that is why we call them obligate parasites. As I already said earlier, this is aseptate. If it's aseptate, then obviously it will be cenocytic, right? Now let's move on. How does asexual reproduction take place here? It takes place either in the form of asexual zoospores or in the form of aplanospores, which are nothing but sporangiospores. This is another name for sporangiospores. We've already discussed these two types of spores. Zoospores are motile flagellated, produced inside structures called zoosporangia, so they are endogenous. Aplanospores are also called sporangiospores. They are non-motile but produced inside sporangia, so they are also endogenous. Both of them are endogenous. This is motile, this is non-motile. Sexual reproduction happens by the formation of what we call the zygospore. Remember, I told you that within phycomycetes you have two groups, right? Zygomycetes and oomycetes. Actually, in zygomycetes, the type of spore that forms is zygospore, and in oomycetes, the type of spore that is formed is called oospore. There is a little difference. Okay, if you remember, when we did sexual spores, three types, I told you about oospore, ascospore and basidiospore. And here there is a mention of zygospore. right? So in the same chapter, in one place they've mentioned oospore and in one place they've mentioned zygospore. Both oospore and zygospore can be seen as sexual spores that are formed in the phycomycetes group. Okay. So this zygote formation, when zygote formation happens, zygote itself will develop a thick wall and form the zygospore. That's why it's called the zygospore in zygomycetes. So this fusion of gametes can happen in one of three ways. It can be isogamous, anisogamous or oogamous. Iso means similar. So in isogamy, both the gametes will have similar size and morphology, as in the case of this. You can see they are similar in size and morphology. In anisogamy, one is large, one is small. In oogamy, the female specifically is large and non-motile. The male is small and motile. Okay, it can happen in any of these ways. Different species, it happens in different ways. Okay, now let's talk about ascomycetes. I've already told you that ascomycetes are called sac fungi because here there is a sac-like structure that is formed called the ascus. It is within that ascus the spores will form. Okay, that's why we call it the sac fungi. Generally, these are multicellular, but unicellular yeasts also belong to ascomycetes. Okay. Multicellular example, penicillium, yeast, Saccharomyces cerevisiae is an example for unicellular ascomycete. Like most fungi, they are saprophytic decomposers. Some of them could be parasitic. One important point here, some of them are coprophilous. That means they grow on fecal matter. They grow on dung. Mycelium here, we obviously know in the next three groups in ascomycetes, basidiomycetes and deuteromycetes, we know that the mycelium is branched, it is septate. Okay. Now, how does asexual reproduction happen here? Asexual reproduction happens through the formation of conidia. What conidia is? I've already told you. These are conidia, they are non-motile spores. They are pinched off of the tip of structures called conidiophores. At the tip of this, they get rounded off and released. Okay. So, asexual reproduction here happens by means of conidia. How does sexual reproduction happen? Sexual reproduction here happens by means of ascospores. Ascospores are produced inside structures called ascus. Ascus is singular, asci is plural. Okay, so here you're looking at these sac-like structures that I'm going to outline in red right now. These sac-like structures are called asci. Singular is ascus. Okay, so within this ascus, you can see the formation of these pores. Okay, so these are called ascospores. Within each ascus, there will be either 4 or 8 ascospores present. Now, these asci are present inside fruiting bodies that we call ascocarps. Ascocarp. Carp means fruit. Okay, so this is a fruiting body. Below the surface of the substratum, you will find vegetative mycelium. During sexual reproduction, especially in ascomycetes and basidiomycetes, you can see that the dikaryon phase is much long-lived. The dikaryon phase itself will give rise to this ascus and everything. Okay, so here you can see these are the asci right here. Okay, it's within these asci that ascospores are formed. Alright, now here are the examples of ascomycetes. Aspergillus, claviceps, neurospora, these are examples. Neurospora is used in a lot of research related to genetics. Okay, so it is used extensively in biochemical as well as genetic work. And just like mushrooms are edible, the fruiting bodies of ascomycetes, the ascocarps, also are considered to be edible. So those ascocarps are available in the form of morels and truffles. It is considered to be a delicacy in many parts of the world. Okay, that's about your ascomycetes. Now let's talk about basidiomycetes or club fungi. most commonly known type of basidiomycete is mushroom. Along with that, you have fungi called bracket fungi. You may have seen this. Okay, mushrooms have this umbrella-like structure. These are also belonging to the same group, basidiomycetes. These are called bracket fungi, and you have these kind of fruiting bodies formed in some species. These are called puff balls. Okay, where do they grow? Similar to most other fungi, they grow on they grow in the soil. They could grow on logs, stumps of trees. In living plant bodies, they can grow as parasites and cause diseases. Okay, the types of disease that basidiomycetes cause, some examples of that are rust and smut. Rust I had already shown you, right, what it looks like in the wheat plant. So the mycelium here, like we already know, it is branched and septate. It is aseptate only in phycomycetes. In the other groups, it is septate. Okay. Now, how does asexual reproduction happen here? Here spore formation will not happen, but you can see fragmentation occurring. This does not have any specialized sex organs. Okay, talking about how sexual reproduction happens here, you can see there are two opposite mating types. We'll start from here. So here is plus, here is minus. These are the two opposite mating types. You can see how one cell from here and one cell from here, direct somatic cells themselves have fused. First, plasmogamy has happened. After that, you have dikaryon stage. I told you in ascomycetes, basidiomycetes, dikaryon stage will persist for a really long time. So, in the dikaryon phase itself, you can see the formation of the fruiting body, which we call the basidiocarp. Inside the basidiocarp, you will have structures called basidia. Okay, so these, I will draw basidia here and show it to you. If this is the basidium singular, this basidium produces haploid basidiospores, but they are not produced inside the basidia like inside the ascus the spores were produced. There it was endogenous. Over here it will be produced outside. Okay, so these are called basidiospores. Once these basidiospores are released, it will germinate and give rise to hyphae again. Okay, so in the dikaryon phase itself, basidia will form, and within the basidiocarp, karyogamy will happen, basidium will form. In the basidium, I mean on the surface of the basidium, you can find 4 haploid basidiospores formed. Once they are released and they germinate, you will find hyphae or new mycelium will form. Okay. Plasmogamy is brought about by fusion of 2 vegetative or somatic cells of different strains. The resultant structure is dikaryotic. That resultant structure, the dikaryotic structure itself, will give rise to the basidium. In the basidium itself, nuclear fusion, karyogamy and meiosis will take place. As a result, 4 basidiospores are produced. The basidiospores, when they germinate, it will give rise to new hyphae. Okay. Some examples of basidiomycetes. Most popular example is agaricus or mushroom, eustallago or smut, Puccinia, which causes rust disease. Okay, this is what a smut will look like, that is mushrooms. Okay, now let's talk about the last group, which is deuteromycetes. They are called imperfect fungi because apparently sexual phase is called the perfect stage. In these organisms, either they do not undergo sexual reproduction or we have not yet discovered how they undergo sexual reproduction. As of now, we only know that they undergo reproduction by asexual means. As and when they've discovered how they reproduce sexually, depending upon what kind of spore they produce, they will classify it into either basidiomycetes or ascomycetes. But this includes organisms in which sexual reproduction doesn't take place or we have not yet discovered it. Okay, so sometimes what happens is the same species will be grouped into 2 different groups thinking they are different species. Eventually when they find out, it will be moved back into the group that it belongs to. Okay, so they are commonly called imperfect fungi. The reason is because only the asexual or vegetative phase is known. When sexual forms are discovered, they will be moved into other classes. It is also possible that asexual vegetative has been given one name and called one species, and sexual stage is given another species. Later, whenever they make the connections, they will move it accordingly. Okay. So yeah, not much is given about the deuteromycetes. How they perform asexual reproduction is by asexual spores called conidia. Mycelium, like we already know, it is septate and branched. Some of these organisms are saprophytes or parasites. Many of them are decomposers. They are involved in the breakdown of detritus, litter. If they are involved in breakdown, and obviously they will help in the cycling of minerals and nutrients. Examples for these: Alternaria, Colletotrichum and Trichoderma. You have to remember this. Now, here is an overall comparison of different types of spores, different ways of reproduction that will happen in fungi. In phycomycetes, what are the asexual spores formed, sexual spores formed, and what is the fruiting body called? Does it have a fruiting body at all? All of these is mentioned here in this table. You can take a look at it and refer to it in your notes. Okay. Now, here is another slide that I've added listing down the examples of each group of fungus. So, you can use this to revise in your notes. Now, that's about kingdom fungi. We have to learn about kingdom plantae. There's an entire chapter dedicated to plant kingdom, which we will learn about next. So, for now, let's understand overview of what plant kingdom includes. In detail, we will learn in our next chapter called plant kingdom. So, what do we already know about plant kingdom? We know that they are eukaryotic, we know that they are multicellular, we know that they are autotrophic or photosynthetic, right? So, they are eukaryotic. If they are photosynthetic, then 100% they will contain chlorophyll. Without chlorophyll, photosynthesis will not take place, right? Some of them are called partial heterotrophs. Examples for that are these insectivorous plants. These two are insectivorous plants. Okay, so this is called bladderwort. This is called Venus flytrap. Basically, they are called partial or semi-heterotrophs because they have leaves like normal other plants. They are capable of photosynthesis. Plants require nitrogen to make their protein, to make their DNA. These organisms, these plants grow in soils where nitrogen is not sufficient. So, they have mechanisms to trap insects. They will trap the insects, and from the insect, they will absorb the nitrogen. Okay. So, they are capable of photosynthesis, but they are dependent on insects because of nitrogen deficiency in the soils. Okay. That is why they trap insects. Very few species of parasites also you can see. Cuscuta is an example. Cuscuta, it is non-green. It is not green in color. It's not capable of photosynthesis. It will depend on host plants like hedge plants. What it does is it will produce specialized roots, parasitic roots called haustoria, and it will penetrate into the host plant to the phloem, so it can directly derive nourishment from the host tissue itself. It will directly penetrate the vascular tissues and obtain nourishment from it. So, such parasites also you can see in plants, but it's very rare. We already know the structure of a plant cell. It's eukaryotic. It will have a cell wall that is made up of cellulose. Also, for photosynthesis, chloroplast will be present. Under plant kingdom, you have different groups like algae, bryophytes, pteridophytes, gymnosperms, angiosperms. More about it we learn in plant kingdom in detail. Also, one characteristic feature of plants is you see something called alternation of generation, where a haploid gametophytic phase will alternate with a diploid sporophytic phase. Again, more about that we learn in plant kingdom. So, for now, just remember this. Now, let's talk about animal kingdom. So, what do we already know about kingdom Animalia? They are multicellular organisms, they are heterotrophic and eukaryotic, right? They show holozoic mode of nutrition, which is nothing but ingestive heterotrophy. We call it holozoic. They are multicellular, heterotrophic, eukaryotic. Their cells don't have a cell wall. They are heterotrophic, directly or indirectly they depend on plants for food. Yeah, and how they store their reserve food is in the form of either glycogen or fat. More about animal kingdom you will learn in the chapter animal kingdom in zoology. Okay. So, they have a definite growth pattern. They do not grow forever like plants, indefinitely like plants. In a plant, if a leaf falls off, a new leaf will grow. But if you cut your arm will not grow. So, it is, they follow a definite growth pattern. And if you look at the adults of animals, they will have a definite shape and size. Okay. In higher advanced animals, you can see very advanced, elaborate sensory as well as neuromotor structures. More about it you will learn in the human physiology chapters. Okay. And majority of them are capable of locomotion. They have locomotory structures. Sexual reproduction happens by mating copulation between male and female. Following that, embryological development also will take place. Okay, so that is about your animal kingdom. More about it you will learn in the chapter Animal Kingdom. Now, there are a couple of organisms which are acellular. You cannot group them in either of any of the 5 kingdoms that we've discussed so far. One example of that is viruses. Viruses, unlike other organisms we've discussed so far, lacks a proper cellular structure. It does not have like a cell wall. It does not have organelles of its own. All it is made up of is its genetic material, which could either be RNA or DNA. And along with that, it will have a protein coat to protect it. Okay. So viruses are non-cellular organisms. They are obligate parasites. Okay. They are obligate parasites, which means in the absence of host, they cannot survive at all. They are equal to non-living things. Whenever they are living outside the host cell, they will have inert crystalline structure. As soon as they infect their host, they will take over the cellular machinery of the host. They will use the host enzymes, they will use the host organelles and make more copies of themselves. Alright. So, virus means venom or poisonous fluid. Generally, viruses cause so much harm that it kills the host itself. That's why it's apt to call them venom, right? So, it is poisonous. That's why it will kill the host, right? So, that's why these organisms are called viruses. Now, how do we know about viruses? Because of the research of certain scientists. One such scientist is Dmitry Ivanovsky. In the year 1892, he was studying about a disease that was caused in tobacco plants. It's called the tobacco mosaic disease. In the tobacco mosaic disease, you will see mosaic-like pattern on the leaves. The leaves will start losing chlorophyll, yellow colored patches will start appearing. That is a symptom of the disease. So, when Dmitry Ivanovsky was researching about tobacco mosaic disease, he found out that whichever organism was causing this disease was not a bacterium. It was smaller than a bacterium because it could pass through the filters which bacteria could not pass through. Okay, so there are filters with very small pore size which bacteria cannot pass through, but these organisms are much smaller. They can pass through even those bacteria proof filters. So that is his contribution. Next, we have M.W. Bejanek. What he said was, he took a infected tobacco plant. He took the extract of that and introduced that into a healthy tobacco plant. The healthy tobacco plant also got the disease. So, he called that fluid Contagium vivum fluidum, which means infectious living fluid. And he named this new organism or the new pathogen as virus. That is his contribution. Then we have W.M. Stanley. So, he is the person to have shown us that viruses can be crystallized outside the host, and generally these crystals are majorly made up of proteins. Now, let's talk about the structure of viruses. I already told you that they are obligate parasites. They are made up of just 2 components. It is a nucleoprotein, which means nucleo means it has nucleic acid, either RNA or DNA. Protein, nucleic acid in the center. Surrounding that, it has a proteinaceous outer coat or covering to protect the genetic material. Now, this protein coat that is there outside has a special name. We call it the capsid. Now, this capsid has many subunits present in it, which are called the capsomeres. This is a diagram of the tobacco mosaic virus that I spoke about, and I want you to notice how the capsomeres here, each individual unit here is the capsomere, how it is arranged spirally, and within that you can see the genetic material being present. Now, in different viruses, the capsomeres are arranged differently, and so the shape of the virus also will vary. Okay, so these capsomeres can be helical as in this case, or sometimes they can have polyhedral geometric shapes as well. Next, So over here you have the protein and the genetic material. The part which causes the infection or the infectious part is the genetic material. It is not the protein. Okay. So yeah, it is the genetic material that is infectious. No viruses contain both DNA and RNA. It can either contain DNA or contain RNA. Generally, plant viruses have single-stranded RNA as the genetic material. What we saw here is a plant virus, tobacco mosaic virus. It has RNA single-stranded as the genetic material. Animal viruses can have single-stranded RNA or double-stranded RNA or double-stranded DNA. Bacterial viruses, which we call bacteriophages, they usually have double-stranded DNA. Okay, now viruses cause several diseases in animals, such as the recent COVID that was also caused by viruses. We have AIDS, mumps, smallpox, herpes, flu, influenza. All of these are caused by viruses. In plants also it is known to cause many diseases. Usually the symptoms of viral diseases in plants include formation of mosaic on the leaves, curling of the leaves, rolling of the leaves, vein clearing we say, where the veins, wherever the veins are there, their chlorophyll will be lost and it will appear yellow in color. And then the plant will not grow very tall, it will be dwarfing and stunted growth. Okay, that's about viruses. There's another group of infectious agents which we call viroids. Now these viroids were discovered by a person called T.O. Diner in the year 1971. These organisms are even smaller than viruses. This is known to cause diseases in plants. It causes a disease in potato plant called the potato spindle tuber disease. This infectious agent does not even have protein coat, it is just free RNA which is of low molecular weight. Okay, that is about viroids. Then we have something called prions. Prions are also infectious agents that are known to cause diseases in animals. Prions have a similar size as viruses. Viroids are the smallest. Okay, so here there is no genetic material, they are just proteins which are abnormally folded that causes diseases, generally affects the neurological system. Your nervous system is affected. Okay, yeah, these agents are called prions. Now, two diseases you have to remember which prions cause. One is in humans, one is in cattle. Okay, what disease is caused in cattle is called the mad cow disease in common layman language. It's also called bovine spongiform encephalopathy. Something similar happens in humans as well. It's called Creutzfeldt-Jakob disease or CJD. Okay, now we're reaching the last part of the chapter, lichens. I've already mentioned lichens to you when I spoke about fungi and symbiotic association. In lichens, you have algae and fungi in very close association. Okay, and if you look at this organism, you won't even feel like they are two different organisms. This is what it appears like. Okay, so in lichens, the algal partner has a special name. It's called the phycobiont, and the fungal partner is called the mycobiont. Phycobiont, algae being autotrophic, prepares food and gives it to the fungus. In return, fungus being heterotrophic, it will take up the food from algae, and in return it helps algae by giving it shelter and absorbing nutrients and water for the algae. Okay, so lichens are considered to be pollution indicators. That means wherever pollution is there, these will not grow. If lichens are not growing in an area, it means that that area has very high level of sulfur or high level of air pollution, mostly because of sulfur compounds. Okay, and if in an area you see good amount of lichens growing on trees and rocks, it means that the air quality there is good. Okay, so that's about lichens and that's about biological classification. Hopefully this session was helpful. Hopefully you have no doubts in this chapter anymore. I'll see you again with the next chapter, Plant Kingdom, the next week. So until then, keep studying hard, keep learning. Thank you.